genetic recombination in E. coli. It was done in 1958. The experiment is referring to 1958. It was first reported by. Okay, now this type of question you see, if you know the answer already, you can answer. Otherwise, you can't. This is not a mathematics problem. And you can't work out the problem and then give the answer. Either you should know the correct answer already, or when I am giving the right answer, you know the answer and then memorize it. Okay. These things are, otherwise it is not possible. These are all remembrance or recovery or knowledge type of question. If only you know the answer already, you can understand it. Otherwise, you can't solve this problem. Okay. Now, some of you, at least one of you know what is the genetic recombination. It was first reported by the genetic recombination in the E. coli was first reported by. Any idea for you? So Lederberg is the correct answer. He was responsible for reporting the genetic recombination in E. coli. Okay, E. coli. Now Griffith experiment is something different. Griffith experiment is different. You know Griffith experiment, or should I explain to you? Now, Griffith experiment is a very, very important experiment. I will just take 1 or 2 minutes to explain it to you because it's very important and definitely you are going to get a question on that. So, Griffith, what he did, he took two type of bacteria, namely rough type and smooth type. Rough type is avirulent. Okay, it will not kill any, it will not cause the pneumonia disease. Smooth type is virulent and it will be causing the pneumonia disease in the rats or the mice. This is the situation. So when this rough bacteria which is avirulent, when injected into the mice, now mice, I will put here. It killed, so it will not kill, it lived. Because you see, it is an avirulent type. It is not a disease causing type. When you put the smooth one into the mouse, very good, excellent. That is a Griffith. The principle is called a transformation principle. It is called as the transformation principle, and then it died. When you mix these both, it will not be living, and in the fourth experiment, it will be living. So by this he was able to conclude. But one thing I want to tell you, Aravind, see, when he did this experiment, everything was okay, but the conclusion part of that was wrong. What he told was the sugar molecules and the phosphate molecules are responsible for the transfer of the character from one to another. He made a mistake in that. See, while you are transferring, mixing the two type of pneumococcus bacteria, a rough type and a smooth type, when you are mixing this, the genes of both are getting mixed. So in that genes, actually which is getting mixed in the DNA, he came to a wrong conclusion because he also knew that the DNA is formed of phosphate, sugar and nitrogenous bases. All the three are there. He knew that. But he was under the impression phosphate and sugar molecules are doing this job of transferring the character. He did the mistake. But of course, nobody knew that it was a mistake. But of course, Griffith was awarded Nobel Prize for that. But later on only they found out that it is the DNA molecules and not the sugar and the other things which are involved. Sometimes he was also doubting for protein also. So he did a mistake there. Fine. So what I am trying to explain is only Griffith experiment. He already posted, so I am just leaving it. So transformation experiment. You know what is a transduction? You know what is a transduction? Can anybody post me what is the difference between the transformation and the transduction? What is the basic difference? Basic difference between the transformation and the transduction? See, in a bacterium, you take a bacterium, you have got the DNA molecule, circular DNA. Now, when a phage is attacking, when a phage is attacking, This DNA goes inside and then it is a multiplying. Okay. Now by this time this DNA is also getting disintegrated. And then this DNA is mixing with this DNA and then you will be getting a bacteriophage inside with the DNA of the bacterium. So this is a virus but with the genome of the bacterium. Now what happens when this one is infecting this? What will happen? This DNA from this bacterium will be transferred to here. This is what is called a transduction. So the transfer of genetic material from one bacterium to another bacterium with the help of a bacteriophage. If two bacteria come together and if it is by means of a conjugation, then it is called a transformation. So in Pneumococcus bacteria, that's what happened. Two bacteria came together, R type and S type. Then there was a genetic transfer. But here what happens, the two bacteria, they are very far off. Nobody knows which bacteria was there. And the DNA is carried from one bacterium to another bacterium with the help of a phage. That is what is called as a transduction. I think now you have got the point.